So on this little handout, there were some questions, and the questions were fairly straightforward and basic. The answer was very similar for a lot of them. I know a few people asked me, is it really this every time? And I said, yes, it is. It's the same thing. But the idea for all of the questions is that you recognize that to put two small pieces together, I have to take something off of them. So let's say, for example, that I want to take the first two monosaccharides and put them together like it was a puzzle. It would make sense for me to cut off those two little bits. That way, there will be a hole in the bottom of the first one, and there'll be like a little peg on the top of the second one, and they'll fit together like a nice little puzzle. Now, if I want to then attach the third one, it would make sense for me to repeat the same process. Cut off the hydrogen from the second one and the O and the H from the third one. So the idea is that each time you add one piece, you lose one water. And that's how you would answer the questions. What did I have to lose to add the first two together? An H and an O and an H. That's a molecule of water. Now I color-coded them so you could see how they would fit together. So the yellow sides go together, the blue sides go together. Does anyone want to ask any questions about that part? So just so that we're clear, when I put two of them together, it's called a disaccharide. When I have three or more, we'll just put that in the polysaccharide category. I know a few people asked me if they could say trisaccharide, and I said, sure, that is exactly what it would be, but trisaccharide is not really a term that we're going to use. So I said you could go with the generic polysaccharide. Yes? You can just say polysaccharide. It's kind of a general term for several pieces together. Normally it's thousands, but I didn't want to make you guys put 1,000 pieces together to make a polysaccharide. So I stuck with 3. You could specify and say 3-piece polysaccharide. That would be fine. Or you could stick with trisaccharide because that's not wrong. That's exactly what it is. I'm just saying you can be general here and just use poly. Does anyone else have anything that they want to ask about the first part? So on the second part, we're doing essentially the same thing. On the second part, this all together is glycerol. So you don't take it apart. We're imagining that it's all attached together. Glycerol is the backbone, and each fatty acid will get added to glycerol. So I'll color code it again so that you can see where you would cut. I would cut off this part here, that yellow part there. So I've got sort of a little peg. And the H off of one of the fatty acids. Then I could stick them together, right? The peg from the glycerol will fit in the little space on the fatty acid. I would do the same thing on the next one. Cut off the OH here and the H there, so I could stick the next one on. And then finally, this OH and that H. So the idea, just like with the first one, is every time I attach one piece of something, I lose one molecule of water, an H and an OH. So the color coding shows you how they would fit together, yellow with yellow, blue with blue, green with green. The idea here is that I lose 3 waters to make 1 triglyceride fat. So on the daily review, when I asked you what happens when I add 3 waters to a triglyceride fat, I put it back to this, glycerol and 3 fatty acids. All right, so if you have the printed out notes, the next slide was in lesson 3, but we didn't finish it yesterday. So I just slid it ahead and put it in lesson 4. Lesson 4 is all about proteins. What are they like? And we're going to talk about their function. Now, because the function of proteins is one of the things that we focus on in this unit the most, we're going to spend a few days talking just about proteins. So we spent one day talking about carbohydrates and lipids together. We're going to spend like 3 talking about just proteins. So you can see the number of questions would be proportionate to that. So I put as part of the title here, why can't I uncook an egg? Because we're really going to focus on the structure of a protein and the functions that it can perform and what might change the possibility of a protein performing its functions. So, proteins. You'll see at the top the abbreviation or the little mnemonic there. Instead of CHO, like it was for carbohydrate, or CHOP, like it was for a lipid, this one is CHONS. So the C is still carbon, the H is still hydrogen, the O is still oxygen. The N is quite important. The N is nitrogen, and it is really quite key. When you're looking at a diagram of a molecule, if you see N, please pick protein. That is the answer. Carbohydrates do not have nitrogen. Lipids don't have nitrogen. Proteins always, 100% of the time, have nitrogen. The S is for sulfur. You don't always see it as part of the picture, but it's frequently there to hold bits of protein together. So I like to make sure that you know about that one. So proteins are made from those elements. Those elements link together to form what we call an amino acid. So we have these little building blocks. For carbohydrates, it was monosaccharides. For lipids, it's fatty acids and glycerol. For proteins, it's amino acids. In your textbook on page 209, there is a picture much like the one at the bottom that shows you what your basic amino acid looks like. Like most structural diagrams, I'm not asking you to recreate it. I just want you to recognize what it looks like. And I want you to notice there's nitrogen. That is like the key distinguishing feature of an amino acid. Now, there are actually 20 different amino acids that are used to make every protein for every living thing on Earth that we are currently aware of. Only 20 of them. It's quite amazing. If you take Bio20, you learn more about these amino acids. For now, I'm just going to state that they exist. On this picture, you'll see at the bottom there's just the letter R, which isn't actually an element. That piece on the bottom is the R group. That's the variant. That's the part that's different on each amino acid. And for the purpose of our course, I'm not usually going to draw a specific amino acid. I'm just going to leave the R most of the time to say this could be any amino acid because we don't necessarily care which one it is. So an amino acid is the building block of a protein. The amino part of the name, you might see in the picture, comes from this part of the molecule. If you see amine or amino as part of a name, it's something with nitrogen. That's what that prefix goes with. Now on the right, I have a picture of the formation of a peptide bond. Whenever you see pep as part of a word, it very probably means something about protein. So a peptide bond is a bond that's between the pieces of a protein. So it's just a fancy name for a bond between those two amino acids. Now I have a picture here of dehydration synthesis, which we've already seen. And page 211 of your textbook has another diagram should you wish to see a different version or to perhaps recreate the diagram yourself. Just like with the other macromolecules, if I want to make a peptide bond, I'm going to look in the middle. I'm going to find 2 H's and an O that are adjacent to each other. I'm going to remove them. And then I'm going to have a new attachment in the middle where my two amino acids are now joined. The reverse is also possible. Hydrolysis, I can put that molecule of water back in, break the two amino acids apart. So then the next thing is protein functions, and there are quite a few. So I'm going to ask you to do a little summarizing here.